Hi everybody, it's Mrs. Mancuso and I'm going over the question for Monday, May 11th, practice AP Bio. So this is very typical question that you would get for number 1, which has 10 parts. Each part is worth 1 point and each part has one task verb. So one thing that you need to do in it for the point. And then this question, you will have 25 minutes to answer it, 5 minutes to upload it, and it will be worth 65% of your grade. All right, first question. In many countries, this type of mosquito is responsible for transmitting the parasite that causes malaria to people through their bites. A primary tool for mosquito control is the use of insecticidal nets sprayed with chemicals known as Ps, which are relatively safe for people but toxic to mosquitoes. However, mosquito resistance to P has now become widespread. P interferes with the function of a transmembrane sodium channel found in the cells of the mosquitoes. In one common mutation to the channel protein, a phenolphthalein is substituted for a leucine at amino acid position 1014. Scientists hypothesize that this mutation is responsible for some cases of P resistance. Okay, so let's digest what we've read so far. You know that I either leave off words like, I don't need to know the genus species name of this mosquito. If I have to write it in my answer, I will, but there's no sense trying to like read it and not stumble over it. Same thing with the insecticide that's used. I just call the insecticide chemical P. So P kills mosquitoes unless they have this mutation. All right, the mutation is a change. All right, a phenolphthalein is substituted for a leucine. So it changes the P to L at 1014. That's the change. Okay. This P kills mosquitoes because it interferes with the sodium channel. All right, so that is what I have interpreted of what I have read so far. Now let's keep reading. So to investigate P resistance, mosquitoes were collected 4 times over a 2-year period from the following 2 regions. Region A, a southern vegetable growing region where large amounts of insecticide are applied for crop protection. Region B, a northern rice growing region where very little insecticide is applied for rice protection. Okay, so I'm already starting to guess that this area where they already have the insecticide, they may already be resistant. This is just a guess. And these ones are probably not resistant because there's no need for that particular mutation to have become higher in frequency in that area if it does exist at all. All right, so scientists exposed the collected mosquitoes to filter papers soaked in 2 different P insecticides, D and P. And the percent mortality of the mosquitoes was determined after 24 hours. Okay, so basically they take the mosquitoes. So here's my little mosquito, my little flying bug. They put it into a container, and in here is soaked filter paper. So the soaked filter paper, they do some of them with the P, and they do some more with the soaked filter paper with the D. And they're going to do this for region A and for region B. And they determined whether, once they did this and they saw how many, the percent mortality means the percent that die. So once they figured out how many had died, then they figured out if leucine or phenolphthalein was at that position. Okay. So here is our data. Region A is already treated with insecticides and region B had no insecticides. I'm just reminding myself of that. And so what happens here? So I'm guessing that this is the insecticide that they used at the beginning. So these ones are already resistant. And the reason why I say that is they have low mortality, which means most live. These seem to be not very affected over time by this. This one over here from 2008 to 2010, there is not a lot of change. I'm just going to put no change here. No real resistance seems to be happening. Maybe it's like 25% are resistant at the start, but I don't see it really changing a lot over the course of time. All right, now let's go over to region B. So I start off here with the P. So most of them die with the insecticide. Most die when exposed to the insecticide. But then look, this is unbelievable. In the end, most live. So I'm guessing, like, do they become resistant over this 2-year time period? All right, next one, same thing. Boom. I see this great decrease in mortality, so a higher chance of living in region B as long as you have this resistance. Okay, so it says susceptibility of mosquitoes from 2 regions to the P or D to the 2 different insecticides. A mosquito strain that is susceptible displayed at least 95% mortality, and all experiments in mosquitoes exposed to untreated filter papers displayed less than 10% mortality. Okay, this is going to be important. Why did you expose them to untreated filter paper? This is our control. I need to be able to show that the one and only thing that I'm changing in these conditions is my insecticide. So I would have to have one like this that has no P, no insecticide. So that's why they would have untreated filter paper. And then error bars represent standard deviation, and ND means no data was available. All right, frequencies of leucine and phenolphthalein at position 1014. So if you're homozygous for leucine, you're LL. If you're heterozygous, you're LP. And if you're homozygous for phenolphthalein, you're PP. So I'm just making these up. These are the alleles that you have. And so let's just read what that change was. It changed from P to L. Oh yeah, look at this. So these already changed. Oh, it must be from L to P. Did I do that? All right, so phenolphthalein is substituted in one common mutation. It's substituted for a leucine. Okay, I wrote this backwards. It used to be leucine and it's now phenolphthalein. All right, that makes sense. Okay, so these already changed. So these are already resistant. These guys. Oh, and the ones that didn't already changed or changed by 2008 to 2010. So there's nobody left. There was very few that hadn't changed yet. And by the end of the 2-year time frame, they'd pretty much, not every single one, because some are heterozygous, but the majority of them have now changed to be big P, big P. I could actually figure out the allele frequencies by doing my P's and my Q's. All right. Now I have region B. So region B has not been exposed before, or been exposed very little. And so 20 versus 2. Oh, okay. So this is what I would call the original unchanged genotype. And then after the course of time, now we have an increase in the PP genotype after the exposure to the insecticides. Well, actually, they didn't even really get exposed because they're in an area that doesn't really get sprayed. These ones definitely saw a change over time. All right, so describe the most likely cause of amino acid substitution in the channel protein. So that's a mutation. Oh my gosh, my writing is so awful on this. I so apologize. So there is a mutation in the DNA sequence. You could even say that this is a missense mutation. So it codes for a different amino acid. All right, and that is going to So a mutation in the DNA sequence codes for a different amino acid in the sodium channel polypeptide. And the missense is happening at 1014 at that position. So within this sodium channel polypeptide gene, there was a missense mutation at 1014 that changed it from L to P. All right, explain how the substitution of a single amino acid in the channel protein could cause resistance. Okay, so the substitution caused a change. Sorry, let me write this all out. So the substitution caused a change in amino acid of the channel protein, which alters its shape. When it changes its shape, it is now less likely for the insecticide to bind and like interfere with the function. So mosquitoes live. If the shape is now different, the insecticides can't bind anymore. So now it works. All right, identify the dependent variable. So the dependent variable is what you measure in your experiment. So we were measuring percent mortality. Percent mortality is what was measured. Or you could call it susceptibility. Either one is fine, but that's what you measured. And when it says identify, you really just have to write what it is. All right, identify the positive control. So I'm going to make a whole separate video on what positive versus negative controls are, but a positive control is a group that uses a known treatment to get results. All right, so if you had a negative control, you would expect no different results, but a positive control, you're expecting a result. So they tested, so testing a strain that is susceptible to insecticides. So region B is definitely susceptible because it doesn't usually get insecticides, so you know you're going to get a result. So that is your control. All right, justify exposing some mosquitoes to untreated filter paper each time. So this is done as a control to show that filter paper doesn't harm the mosquitoes, and that the death is due to the P, that type of insecticide, PY, or however they spelled that type of, the two types of insecticides alone. So the only thing that's causing them to die is that insecticide that they are giving it. All right, next. Based on the data in figure 2, describe whether mosquitoes from region A or from region B are more likely to exhibit greater evolutionary fitness. Okay, so I want to see which ones are most likely to survive. Most fit is equal to most likely to survive and reproduce in their environment. So if I go up here, who is most likely to survive in their environment? Definitely region A, because region A has lower mortality. All right, next one. Based on the data in figure 2, describe any significant change in the susceptibility of mosquitoes from region B to each of the 2 insecticides over the 2-year period. Okay, so I have to do it for both. So I will do it for the P insecticide first, and it seems like there's little significant change until June 2010. So let's go back to my data chart. All right. So for the P, it just seems like there's this to this, there's a significant change there, but there doesn't really seem to be much of a change. And for D, oh, sorry, hang on, just a second. Only region B, only region B. So I'm not looking here. I'm looking here. I was like, I really thought I looked at it and saw a much greater difference. So this seems to be no significant change. And then a huge difference right here. So from October 2009 to June 2010, way less dye. All right. So little significant change until June 2010, then greatly reduced mortality. All right, for the D insecticide, which I would obviously write out the name, but let's look at this one. There is a decrease here and a decrease here. So there's a significant decrease in mortality from June 2009 to October 2009. And then another significant decrease in mortality from October 2009 to June 2010. All right, moving on. Use the data in table 1 to calculate the frequency of the allele coding for phenolphthalein in each population of mosquitoes. Okay, we are going to do this just like we did in our lab. So if these guys are PP and these guys are LP and these guys are LL, I want to do this. Okay, I have to do it for both areas, and I do it only for October 2008. So October 2008, I am just going to look at these numbers, okay? So what I have to do is I have to go down here and I have to say, okay, for region A, I need to figure out the number of P alleles over the total alleles. And that's going to give me the frequency of the P allele. Okay, so the number of P alleles. All right, I've got 31 times 2, because remember, these guys have 2 Ps. So 31 times 2 is equal to 62. Plus, I've got 5 more that are just single Ps because the other letter is a L. So that's equal to 67 over, all right, total number of alleles. So I've got 39 and everybody has 2, so that's 78. So I want 67 divided by 78, and it tells me to do it to 2 decimal places. So 0.86 is the frequency of the P allele. And then for region B, I need to do the same thing. So I have region B is right here. So 2 times 2 plus 5 of them. So that's 9 over, how many total are in this population? 27. 27 times 2 is 54. So 9 divided by 54, much lower frequency of this allele because they haven't been exposed to the insecticide. So 0.17 is the frequency of the P allele in region B. All right, next question. Using mosquitoes from insecticide-free areas, the scientists developed mosquito strains with amino acid substitutions at other positions in it. Predict the susceptibility of the mosquitoes to the insecticide. Okay, they are 100% susceptible or they will die. All will die because you're giving it an insecticide. So my assumption is always that they're going to die if you expose them to an insecticide. And based on the data in table 1 and the information provided, provide evidence to support the scientist's claim. So the claim was that the scientists claim that the mosquito population of region B evolved resistance over the period of the experiment, and the resistance arose as a result of the immigration. Okay, I would totally agree because look at what happened. So you got to say my evidence is that what region B was not treated with insecticides, but did develop resistance. So the reason may be mosquitoes that are resistant from area A or region A immigrate to region B. And as they mate with mosquitoes in region B, they pass on their genes and the frequency of the P allele increases. All right, there you go. Write down any questions you have and I would be happy to go over it.